and I just want to say a massive thank you to the AMF. I'm honestly so humbled and so honoured to be able to take part in such an incredible conference. My name is Gemma Owen, and I am in my... Thank you. My name is Gemma Owen, and I'm in my second year of my PhD at the University of Oxford, where I'm funded by the AMF, under the supervision of Dr. Shiji Kai and Professor David Kerr. And today, I will be going through a very brief snapshot of some of our research, discussing the role of normal cholangiocytes in regulating cholangiocarcinoma cells. So typically, cancer starts by one cell acquiring mutations, which allows it to continue growing and ignoring the signals to stop, a bit like a car with a broken brake pedal and the accelerator pedal stuck down. This abnormal cell does not grow alone. The tumor microenvironment plays a substantial role in modulating the cell motility, growth, invasion, and metabolism. And the microenvironment consists of metabolites, cytokines, growth factors, and extracellular matrix, as well as the cells that provide these. The importance of the interaction between cancer cells, stromal cells, and the tumor microenvironment is well established. However, this can often ignore the interaction with surrounding normal cells. There are multiple levels of reciprocal communication that occurs between the cancer cells and the normal cells. Both direct and indirect signaling from the cancer cells to the normal cells, as well as from the normal cells to the cancer cells. And it is this crosstalk between the normal cells and the cancer cells that we are particularly interested in. We hypothesized that during the early onset of cholangiocarcinoma, normal, non-cancerous cholangiocytes lining the bile ducts play a role in restricting cholangiocarcinoma growth via a shift in gene expression and metabolism. This shift eventually leads to a tipping point whereby the cholangiocarcinoma cells begin to outcompete and are no longer restricted by the cholangiocyte cells. And we believe that the molecular mechanisms in this early interaction can provide clues to the later interactions. So how are we doing this in the lab? We primarily use a 2D indirect co-culture model. So we use an intrahepatic cholangiocarcinoma cell line called CCSW1, as well as a normal cholangiocell line called MMK1. And these are grown together, separated by a semi-permeable membrane, in order to elucidate the soluble molecules during this crosstalk. I touched on it briefly before, but my research aims are to evaluate the effects of non-cancerous cholangiocytes on cholangiocarcinoma cell proliferation and migration, as well as the molecular mechanisms that regulate this crosstalk, with a shift in metabolism and gene expression. And this is with the aim to use this information for the development of biomarkers and potential therapies. To do this, we are using a combination multi-omics approach, combining metabolomic analysis with RNA sequencing to identify common modulated pathways. Firstly, when cholangiocyte cells are in co-culture with our cholangiocarcinoma cells, we see a significant decrease in apoptosis. Apoptosis is also known as programmed cell death, and this is where cells self-destruct if they are too old or damaged. It's a normal process, and it occurs during early development to get rid of unwanted cells, such as cells between the fingers of a developing hand. This decrease in apoptosis occurs in conjunction with the inhibition of a p53 pathway, shown here by a decrease in p53, p21 BAX, as well as a decrease in caspase 3 and caspase 8. When we view the effect of co-culture on the cholangiocarcinoma cells themselves, we see a significant decrease in the amount of transwell migration, with no significant effect on the proportion of migrating cholangiocyte cells. This occurs in conjunction with the inhibition of EMT protein expression, which should be shown here in the Western blot, as a decrease in vimentin and slug expression and an increase in E-cadherin expression. Do take my word that the Western blots are there, and they were definitely there when I sent in the presentation. Epithelial mesenchymal transition, or EMT, is a process whereby cells lose their cell-to-cell adhesion, and they gain migratory and invasive properties. When cells lose the molecules on their surface that keep normal cells in place, they can break away and spread to other parts of the body, which is part of how cancer cells can spread around the body. In order to explain the molecular basis for this decrease in apoptosis in our cholangiocyte cells, as well as this decreased EMT in our cholangiocarcinoma cells, we first conducted a metabolomics analysis to assess the metabolic profile of the cells when alone and in co-culture. And here we just looked at all of the intracellular metabolites across 6, 24, and 48 hours. The heat map on the left identifies the differentially expressed metabolites, and the color of the cell is dependent on the adjusted p-value and the fold chains. So essentially, the brighter the color, the more differentially expressed the metabolite. Using this, we can see a dynamic change in metabolite concentration over 48 hours. And the volcano plot in the middle shows the metabolites differentially expressed at 48 hours. Two that I've highlighted here, choline and creatine, were significantly upregulated at 48 hours, occurs in conjunction with a decrease in glutamate at 48 hours. Now, 2-oxoisopentanoate is involved with valine degradation. And we know that glutamate and glutamine, which glutamine is synthesized from glutamate, can be used to fuel the TCA cycle in cancer cells. We then extracted RNA for genome profiling using RNA sequencing to identify differentially expressed genes and genetic pathways, which we are in the process of combining with our metabolic profiling to see if we can elucidate what's responsible for this decreased apoptosis and decreased EMT. We then conducted a Geneset enrichment analysis using the whole Geneset database to determine the significantly modulated gene pathways. And when we looked at the MM and K1 cells, we saw significant enrichment in pathways associated with the cell cycle, such as the G2M checkpoint, as well as a decrease in apoptosis and P53 pathway signatures. The volcano on the right highlights some of the most significant differentially expressed genes. For example, two I've highlighted here is NPPB, which is a cardiac hormone, and txnip, which is involved with regulating oxidative stress within the cell and also functions as a tumor suppressor. As I said earlier, we're currently in the process of combining these two big data sets, and we're really excited to be able to identify the common modulated pathways. When we look at the gene pathways in our CCA cell line, we see an increase in gene pathways associated with apical junction formation, which is in agreement with our previous work showing inhibition of EMT and reduced migration. What's more is when we look at the individual differentially expressed genes, we can see a stark increase in the fold of txnip, the tumor suppressor that I mentioned earlier. So what exactly is txnip, or thioredoxin interacting protein? We kind of want to know, why is it upregulated in our cholangiocyte cells and our cholangiocarcinoma cells under co-culture? So first of all, what does it do? Well, it's a regulator of cellular redox in the cells, and it protects them from oxidative stress. There are many publications that suggest that the inhibition of txnip via microRNAs can promote the tumor growth and stimulate breast cancer EMT. This occurs via the increase of HIF1 alpha and twist, and this microRNA txnip twist signaling axis is associated with worse outcome in breast cancer patients. It can also promote the proliferation and migration of pancreatic ductal adenocarcinoma. Not only this, but txnip is also modulated epigenetically. And it has been shown in renal cell carcinoma that HDAC1 can be recruited to txnip promoter to decrease txnip expression to promote tumor development. Like what's more, in esophageal adenocarcinoma, entinostat, which is a HDAC inhibitor, can upregulate txnip and synergistically increase cisplatin-mediated DNA damage and apoptosis. What's more is in acute myeloid leukemia, JQ1, which is a bromodomain 4 inhibitor, can reduce the expression of c-MYC, activating the ASK1 MAP kinase pathway via txnip, leading to more apoptosis. These previous published works suggest that our hypothesis, txnip, is upregulating cholangiocytes to protect them from oxidative stress. The opposite is true in the tumor, and that txnip suppresses tumor growth. As such, we're in the process of validating our RNA-seq data using qPCR, Western blotting, and sRNA knockdown. But following on from this, we have also conducted a drug screen on our cholangiocarcinoma cells using an epigenetic inhibitor library. So our epigenetic inhibitor library is a unique collection of 63 epigenetic modulators, including histone deacetylase inhibitors, histone methyltransferase inhibitors, and bromodomain inhibitors. Using our library, we assessed cell viability after 48 hours of treatment, with little to no change in viability shown in black and red, and a viability change of greater than 0.5 shown in green. The heat map on the left shows all of the compounds, and the bar chart on the right is highlighting the 6 compounds that are highlighted in green. DMSO was used as our control, and 5-FU was used as a positive control. And as you can see, the 6 epigenetic inhibitors here performed much better than 5-FU and successfully decreased cell viability. Previous publications, such as in 2016, showed that pan-HDAC inhibitors can synergize with cisplatin and induce cell growth inhibition in cholangiocarcinoma cells. This also further supports the use of epigenetic inhibitors as a novel future therapy for the treatment of cholangiocarcinoma. And we are currently planning to evaluate the IC50 of these compounds in our cholangiocarcinoma cell lines, as well as in our co-culture model. In summary, non-cancerous cholangiocytes play a role in restricting cholangiocarcinoma cell growth during the early onset of cholangiocarcinoma via a shift in metabolism and gene expression. MMK1 cholangiocyte cells experience a reduction in apoptosis mediated by a reduction in p53 protein and gene expression when in indirect co-culture with intrahepatic cell line CCSW1. And this indirect co-culture with MMK1 causes inhibition of cell migration in CCSW1 cholangiocarcinoma cells in conjunction with a reduction of EMT protein expression and an upregulation in apical junction gene signatures. The interaction between normal cholangiocyte cells and cholangiocarcinoma cells is a newly emerging field, and a lot of questions still remain unanswered. However, as our future work reveals the molecular mechanisms involved during this interaction, it is hoped that we will have a new perspective and dimension for cancer detection, prevention, and treatment. We are currently in the process of validating our functional and molecular work with the intrahepatic cholangiocarcinoma cell line CCLP1, as well as and aim to complete these validation works in the intrahepatic cell lines SNU 1079 and perihilar cell line SNU 1196. Likewise, we are currently establishing a 3D direct co-culture model with our preliminary data shown on the right to better mimic the cholangiocarcinoma growth in vivo and further validate our 2D studies. We've also scheduled to conduct clinical correlation analysis using immunohistochemical staining in patient samples, and we'd hope to further conduct tumor mouse xenograft models to validate this further. In a sense, our 2 cell lines are in a cellular game of chess. Each side is trying to win, each side is trying to protect their king, all while unsure about the other move that the other opponent will make. However, if you're a chess fan, you'll know there are lists and lists of popular opening moves. These are the first of a series of actions that will determine how the middle and the end game play out. We are, in a sense, trying to evaluate the opening moves of our 2 cell lines. And this kind of represents the opening moves of cholangiocarcinoma development. If we can figure out these opening moves, we can estimate the pathway and signal shift during the middle and end game during the cholangiocarcinoma development, and in the future, perhaps exploit these pathway weaknesses as a way to develop targeted therapies and biomarkers for the treatment of cholangiocarcinoma. Firstly, I must say a massive thank you to my supervisors, Dr. Shiji Kai and Professor David Kerr, as well as a thank you to everyone in my lab and the supporting labs. And I can't thank the AMF enough for all of their help, and this research really is entirely about them. So